Hello and welcome to Chapter 15. As this picture actually shows, we're going to talk about the specific or adaptive immunity, and that's the immunity that you are born with or that you acquire after birth. And so I chose this picture simply because do you inherit your immune system? And yes, you actually do. So let's talk about the adaptive response. So specific, acquired, or adaptive immunity. This is the product of the B and T lymphocytes, and each one of them have their own specific functions within the adaptive response. Lymphocytes undergo a selective process that specializes them for reacting to only one specific antigen or immunogen. An immunocompetent patient or immunocompetence is the ability of the body to react with countless foreign substances. So this is why this is acquired, because you acquire it after birth. It's specific to each individual because of what each individual is exposed to. So what are antigens? Well, they're also known as immunogens, and these are chemical substances that stimulate a response by the T or B cells. So it's anything that will cause an adaptive response. Typically, antigens are proteins, but they can be polysaccharide molecules or others. Any exposed or released protein or polysaccharide is potentially, therefore, an antigen. Normally, our own antigens do not evoke a response from our immune system. We actually say that we have tolerance to our own self-antigens. Because the adaptive response is unique to each individual, it does have specificity. It's highly specific to the antigens against which the third line of defense is directed, and that is to those things that the individual has been exposed to. The other thing that separates the adaptive immune response from the innate responses is the fact that we develop memory. This is the rapid mobilization of lymphocytes that have been programmed to recall their first engagement with the invader and rush to the attack again. This allows us then to have a more rapid response if we see this antigen a second or subsequent time. The principal stages of immunological development and interactions include the lymphocyte development and differentiation into the B and T cells themselves, presentation of antigens by a group of cells known as the antigen-presenting cells, and the challenge of the B and T lymphocytes by these antigens. We'll see a B lymphocyte response, which is the production and activities of antibody proteins, and the T lymphocyte response, which is the cell-mediated side. So lymphocytes, both lymphocytes will develop or will begin life in the bone marrow. Once they are mature, they will then be presented with antigens, and depending upon whether they're B cells or T cells will determine then which response that we actually have. So, both B cells and T cells begin life in the bone marrow, but they receive their specialized training or maturation in different areas. B cells will stay in the bone marrow and receive their specialization there, and then travel to secondary lymphoid tissues out in the body. While T lymphocytes are made in the bone marrow, they actually travel as immature cells to the thymus gland and receive their specialized training and receptors there, and then go out to secondary lymphoid tissues or circulate within the blood. The antigen is first processed by a phagocytic cell, and we'll talk about the three types of professional phagocytic cells here in a moment. These are going to be involving the major histocompatibility self-antigen markers, and again, we'll talk about those in a moment. But this antigen-presenting cell will actually engulf and process the antigen and then present that antigen of the foreign substance or the abnormal substance to T cells. The T cells then will actually do a couple of things. They will send out chemical signals to other cells, one of which will be the B cells. Most B cells require stimulation from the T cells themselves. Once activated, the B lymphocytes then become what's known as the antibody-secreting plasma cells. These cells will secrete our antibodies against the specific antigen. While some of the activated B cells will become B memory cells to remember this antigen, and others will actually become something known as the regulatory B cell. The activated T lymphocytes can become several subpopulations of T cells, and the helper T cell will actually form a number of these. But one subpopulation they will form will be the memory T cells. So we make both B and T memory cells. Antibodies produced by the plasma cells will then enter the bloodstream or other body tissues and interact directly with the antigen. And the cell-mediated immune response will include functions of the T helper cell, which I call the general of the immune response because they are helping other cells to become activated. Some will become T regulatory cells that, along with the B cells, will help regulate or actually stop the immune system once the antigen is dealt with, and then other T cells will be activated that are known as cytotoxic T cells, and these are the killer cells. So, major functions of the immune system markers are what we refer to as our self-antigens. These attach to either non-self or foreign antigens and are then presented to the T helper cells. These bind to the cell surface receptors that indicate self, and these are known as the major histocompatibility or MHC molecules or proteins. These markers will receive and transmit chemical messages to coordinate the immune response, and they may actually aid in cellular development. So what are the classes of the major histocompatibility proteins? Class 1 MHC proteins are actually coded for by a group of genes on chromosome pair number 6, and these code for protein markers that appear on all of the body's nucleated cells. So only the mature erythrocyte and perhaps the platelet, since it is not actually a full cell, will not have these self markers. These class 1s actually display the unique characteristics of self. And so these literally are our self markers found on all our cells except again for the erythrocyte. The ABORH blood grouping proteins are going to be what act as the self markers on our red blood cells. And these MHC class 1 proteins allow for recognition of self and the regulation of the immune reaction. Each human inherits a particular combination of these class 1 MHC genes, and what this means is then that their self markers are unique to them. The class 2 MHC genes code for immune regulatory markers. These are found on the immune cells, specifically the macrophages, dendritic cells, and B lymphocytes. These are involved in presenting antigens to T cells during cooperative immune reactions. And there is a class 3 MHC proteins that encode proteins involved in complement system, but we're actually not going to talk about the class 3 MHC proteins. So, class 1 MHC molecules, again, found on all nucleated human cells. However, they are capable of presenting an antigen to a T helper cell. The class 2 MHCs found on certain types of white blood cells, including the macrophages and the B lymphocytes. And by the way, these cells will also have class 1 because they are nucleated cells. So, what class of MHC do we see? Where is it found, or what found on the membrane of what cell? What are their primary functions? Is it is recognized by what receptor of T lymphocytes? And recognition receptor is found on the membrane of what cell? So, class 1s, again, found on the membrane of all nucleated body cells. In the immune system, they actually play the role again of self-antigens, but class 1s will be used to present endogenous or the self-antigens. They are recognized by what's known as the cluster of differentiation, or the CD8 plus receptor found on the cytotoxic T cells. Class 2s found on those specialized immune cells, including the lymphocytes and the professional antigen-producing cells, present exogenous antigens, those that arise from outside the body. They are recognized by the CD4 plus receptor found in the membrane of the helper T cell. So the cluster of differentiation, these markers are important in immunity. They are found on the membranes of a variety of different cells involved in the immune response. There are close to 400 of these CD markers that have been described. We also will find on the surface of the T and B lymphocytes something we call the uh, B and T self markers. And these are basically usually some sort of similar molecule to antibodies. And in fact, in the B cell receptors that we see, these are actually one of the classes of our immunoglobulins or antibodies. And these are actually of the IgD class antibodies. The protein actually sits down into the membrane, and the antigen binding sites will then be exposed on the surface of the cell. In the T lymphocytes, they also have a T cell receptor. It's in a little different shape than the antibody, but same concept. These T cell receptors will actually be embedded into the cell membrane and then stick above with the antigen binding site. And so both the T cell and the B cell receptors are simply waiting for a antigen to be presented to them. Now, tissue macrophages ingest the pathogens and induce an inflammatory response in the tissue if that's an appropriate response. Tissue dendritic cells ingest the antigen, digest it, present and process the antigen, and then migrate to the nearest lymphoid organs. These process and present the antigen then to T helper lymphocytes. So there are three classes of professional antigen-presenting cells: the dendritic cells, any macrophage, and even B lymphocytes can play this role. There are other cells in the body that may be able to present antigens to T helper cells, but that is not their primary function. Whereas with the dendritic cells, macrophages, and the B lymphocytes, this is actually part of what they do on a regular basis. B and T cells proliferate and differentiate when challenged by antigen. So once the antigen has been presented to these lymphocytes, they actually undergo something known as clonal expansion. A clone is simply the proliferation of a particular lymphocyte. In other words, the activated B or T cell that's been challenged by the antigen that now knows the antigen will undergo mitosis and actually present then a huge number of these cells that all know the antigen. These are genetically identical cells, and some of which actually become memory cells. Now, there are three main functions of T lymphocytes. The helper T cells are, again, the generals of the immune response. They, through chemical messengers, will help assist the B cell processes, help activate cytotoxic and regulatory T cells, and activate further macrophages. The regulatory T cells help control the T cell response. We don't want too much of a good thing. So once the antigen has been dealt with, we need some way to downregulate the immune response before it may turn on our own cells. And the cytotoxic T lymphocytes actually lead to destruction of infected host cells, such as cancer cells or virally infected cells, or quote, other foreign or non-cells. B lymphocytes, when activated by an antigens, undergo clonal expansion, giving rise to the three types of B cells. And primarily the main group is going to be the plasma cells. Each plasma cell has the same reactive profile and therefore will produce an antibody to the antigen that B cell was sensitized to. These plasma cells will release the antibodies into tissue, fluids, and blood, and they will be able to interact then with the antigen. The antibodies will actually attack the antigen to which they are specific and mark it for destruction or neutralization. So, antigens are usually what type of molecules? Maturation of B cells occurs in the what, and maturation of the T cells occurs where? And where are both of them made? The MHC marker proteins are found on all cell types except for what? And what are the three professional antigen-presenting cells? True or false? T lymphocytes are the ones that produce antibodies. And true and false. When activated, both T and B cells produce memory cells. So, what are the specific events in T cell maturation? Well, the T cell receptor, or the markers found on the T cells, are antigen-specific T cell receptors. There is a CD3 marker that surrounds the T cell receptor and assists in binding to the antigen for which that receptor has been made. We also find the CD4 receptor on the helper T lymphocyte and the CD8 receptor on the cytotoxic T cells. CD4 again binds to the MHC class II molecules on an antigen-presenting cell, and CDH, found on the cytotoxic T cells, will bind MHC class I molecules. T cells constantly circulate between the lymphatic and the generalized circulatory system. They migrate to specific T cell areas in the lymph nodes and spleen, but if I were to take a blood sample and see lymphocytes, the majority of the lymphocytes in circulation will be T lymphocytes. It's estimated that 1 billion T cells pass between the lymphatic and generalized circulations every day within the body. B cells, while they made in the bone marrow, they mature in the bone marrow as well. Hundreds of millions of distinct B cells develop as the result of gene modification and selection. They circulate through the blood, homing to specific sites in the secondary lymphoid tissue. This is why we find more T cells than B cells in circulation, is the B cells tend to actually go up and take residence in these secondary lymphoid tissues or other areas. They actually have immunoglobulins or antibodies, the class D antibodies to be specific, as their surface receptors. So this table just contrasts the properties of B and T cells. Again, while both are made in the bone marrow, where do they mature or actually receive their specialized receptors? B cells stay in the bone marrow, hence the B for bone marrow. The T cells migrate from the bone marrow to the thymus gland, hence the T cell designation. Specific surface markers in B cells, this is a class D antibody embedded into the cell wall, and we also have some distinct CD molecules. The T cell receptor is kind of a modified antibody structure. It's not a true antibody. And again, we have the distinct CD molecules. B cells tend to be low in circulation while T cells are high. The B cell receptor is again that class D immunoglobulin. The T cell receptor is a modified version. The B cells tend to take up their residence in the cortex of the lymphatic organs, while the T cells tend to be in paracortical sites. B cells do not necessarily require the antigen presenting with the major histocompatibility antigen, whereas the T cells actually do. B cells make plasma cells and memory cells and the B regulatory cells that we're not really going to spend a lot of time with. The T cells will actually divide into several different types of sensitized T cells and T memory cells. The B cells responsible for the production of the antibodies from the plasma cells that will inactivate, neutralize, or target antigens. The T cells function in helping other immune cells, suppressing, killing abnormal cells. They do actually operate in hypersensitivity reactions and can synthesize a whole number of cytokines. Now, each naive lymphocyte bears an antigen receptor that recognizes a unique antigen. Stem cells become granulocytes, monocytes, or lymphocytes in the bone marrow. These lymphocytes actually will then divide or become either B cells or T cells. The B cells will stay in the bone marrow to receive their specialization, while the T cells migrate to the thymus to receive their specialization. And each builds their unique antigen receptor on the surface of the cell. So once sensitized, both B and T cells migrate to secondary lymphoid tissues. Some of these cells, once clonal expansion begins, actually self-destruct if they are not used. The reason for this is they could then potentially start to attack self cells if they are not self-destructed. By the time B and T lymphocytes reach the lymphoid tissues, each one is equipped to respond to a single unique antigen, but they are not yet activated until an antigen is actually presented. So they are basically primed in the bone marrow and thymus in order to receive an antigen, but they are not yet activated. Since they both migrate to secondary, went the wrong way, sorry. So this just shows, again, both B and T cells made in the bone marrow, but the B cells will mature in the bone marrow, T cells will go to the thymus gland. They receive their specialized receptors, and then both will migrate out into the circulation and to secondary lymphoid tissues. Now, the B cell receptor, as I said, is one of the classes of immunoglobulins. And these antibody immunoglobulins are made up of 4 polypeptide chains, 2 heavy and 2 light chains, and we'll talk about those a little bit later on. One of the light chains is bound to one of the heavy chains through disulfide bonds, and the 2 heavy chains are bound together, forming this kind of Y-shaped structure. The variable region will actually contain the antigen binding sites, whereas the constant region down here, which is made up of primarily the heavy chain, is going to identify the class of antibody as well as embed into cell membranes in certain classes. So the antibodies create this symmetrical Y-shaped arrangement of the amino acid chains. Genes that encode for the variable regions will actually make then different protein sequences that are found here in the variable region, and each one of these variable regions will be made to a specific antibody. The antigen binding sites then of the B cell receptor are actually pockets at the ends of the forks formed by the heavy and light chains. These can be highly variable in shape to fit a wide range of antigens. And these variable regions, or within the variable regions, are found the antigen binding sites. The amino acid position is highly variable from one clone of B lymphocytes to another as the result of genetic reassortment. The constant regions, the amino acid content within the C regions, does not vary within a specific class. That's why we can use them to identify what class of antibody we have. T cell receptors belong to the same protein family as the B cell receptor. So they're similar in the fact that they have a variable and constant region. They are actually inserted into the membrane just like the B cell receptor, but they have antigen binding sites that are formed by two parallel polypeptide chains. Unlike the immunoglobulins, T cell receptors tend to be relatively small, and they are never secreted by plasma cells. So, once the lymphocyte stem cell has been undergoing clonal expansion, again, sometimes some of these clones will actually be eliminated through apoptosis. This may be because some of these are actually have receptors that recognize our self-antigens and so would attack self cells. So these ones that would attack self need to be eliminated. The specificity for a single epitope or antigen is programmed then into the lymphocyte and is set for the life of that given cell. The end result is an enormous pool of mature but naive lymphocytes that are ready to future differentiation under the influence of certain organs and or antigen binding. When any antigenic epitope enters the immune surveillance system, it then encounters specific lymphocytes ready to recognize it. Such contrast stimulates that clone to undergo more mitotic divisions and expand it into even a larger population of the lymphocytes, all bearing the same specificity. In other words, if this one is the one that's activated, it will undergo clonal expression and will have a huge amount of clones that bear the same T cell or B cell receptor that can then interact with more of the antigen. Immune tolerance is literally this tolerance of the immune system to our own self-antigens. So that's why any clone of a T cell must be removed that would have a T cell or B cell receptor that could possibly actually bind to one of our own self markers. And we think this may be one reason why autoimmune diseases then develop. So this loss of tolerance may be due to the survival of these, quote, forbidden clones with these receptors for our own self-markers. It may be failure of other systems, or it may actually be loss of the function of the regulatory T lymphocytes, or it may actually be a combination of all three. So the B cell receptor is actually what? True or false? T cell receptors are similar to B cell receptors, except that they are not secreted. True or false? Lymphocyte specificity is pre-programmed. And true or false? B cells and T cells never react to self-antigens. So, how are these antigens presented to the T helper cell? Well, first of all, let's define the antigen again. It is any substance, again, usually a protein, but maybe something else that provokes an immune response in specific lymphocytes. To be perceived as an antigen or immunogen, a substance must meet certain requirements, such as in their foreignness, their shape, their size, and that's an important thing. They must be of the correct molecular size and have accessibility to the antigen-presenting cells. Antigenicity is the property of something behaving as an. So antigens must be perceived as foreign. Well, what actually makes something foreign or non-self? This could be the entire microbe itself or one of their parts, such as a part of the cell wall. So cells are substances that arise from other humans, animals, plants, or various other molecules could all be antigenic. Molecules of complex composition, such as proteins or protein-containing compounds, are more immunogenic than repetitious polymers of a single unit. So, proteins and polypeptides are one of the biggest groups of antigens. But lipoproteins or glycoproteins can also be antigenic. Even some nucleoproteins can be antigenic. And certainly polysaccharides or lipopolysaccharides, especially those found in gram-negative bacteria, could also be antigenic. And so I like this little cartoon because here's our antigen, whatever it is. The antigen presenting cell is going to actually take that antigen and present it to our general over here, the T helper cell. So good immunogens is anything, again, of the correct molecular size, foreign animal or human cells, plant pollens, any of the microbes. There's a whole slew of things that are good antigenic substances. Poor immunogens or antigens tend to be those that are simply something like this, like a trisaccharide or a single polypeptide. Now, again, a substance has to be large enough on a molecular weight basis in order to catch the attention of the surveillance cells in order to initiate the immune response. Molecules with a molecular weight less than 1,000 are seldom complete antigens. Molecular weights of 1,000 to 10,000 are typically very weak antigens, so most antigens need to be of a molecular weight of at least 100,000. Epitope is usually the portion of the antigen molecule recognized and responded to by a lymphocyte. So it may not be the entire protein that the immune system responds to. It may be a small section of that protein. But overall, the protein itself is the antigen. The primary signal that is of a molecule that is foreign is the epitope. Haptens usually consist only of a small determinant group, and by themselves they are too small to elicit an immune response. But if they become linked to a carrier group of some sort, usually found in the host, the combined molecule becomes large enough to develop immunogenicity. So here's our little hapten. By itself, it's too small, basically, to cause an immune response. So we get no immune response whatsoever. But if the hapten becomes bound to a carrier molecule, let's say a protein found in the mouse, then it becomes large enough for the immune system to respond, and we make antibodies. Alloantigens are proteins and other molecules of one person that are antigenic to another person. There are a few people out there that can actually be allergic to other people's epithelial cells or to other cells of the body, and this can actually be a severe problem in those individuals. These alloantigens are cell surface markers that occur in some members of the same species but not in others. This is actually the basis for an individual's blood group and the major histocompatibility profile. So again, for example, I'm type O. That means I would actually be allergic to any type A or type B blood. So I would not be able to receive type A type B or type AB blood as a transfusion because my immune system recognizes the A protein and B protein as being foreign, even though they are human cells. And so this is responsible for the incompatibilities that occur in blood transfusion or sometimes organ grafting with the self-markers. Antigens must further be acted upon and formally presented by, that should say by, antigen-presenting cells. And again, our three types of professional cells are going to be any macrophage, the B lymphocytes, and the dendritic cells. So the first thing that's going to occur is that whatever is bearing the antigen, say this microbe, will be ingested or phagocytized by this, let's say, macrophage. The lysosome will then bind to the phagosome, and we form that phagolysosome. And then what will happen is the antigen or the antigenic epitope will actually be processed and placed upon one of the major histocompatibility antigens that becomes embedded on the inside of the phagolysosome membrane. What this does is when the residual body comes over here and undergoes exocytosis at the cell membrane, that processed antigen will now be on one of the MHC proteins out here on the surface of the cell. So here's our antigenic epitope out here. What this will allow then is recognition of the CD4 receptor on the T helper cell of the MHC2 protein in this case. And what we see is now the antigen is actually sitting between the MHC2 protein and the T helper cell receptor. This then will sensitize this T helper cell to this specific antigen. And what we'll see is that a whole number of cytokines, including some of the interferons and interleukins, will then become involved in the activation of not only the helper cell, but also of other cells of the T helper or of the T cell line as well. So this is actually a dendritic cell. This is the dendritic cell that is in the process of phagocytizing this this fungal spore. So can you list and describe three types of molecules that serve as antigens? True or false, a molecule with a molecular weight of 1500 is strongly antigenic. The blank is the molecule or molecular fragment that signals that a molecule is foreign or non-self. And what is it that is too small to be recognized as a foreign molecule by itself and requires some sort of carrier to be seen by the immune system? T cells respond to what at rates 100 times greater than ordinary antigens. And you may have to do a little research on that one. So once the T helper cell is activated or sensitized to the protein presented by the APC, it then will undergo clonal expansion into several different types of cells. One will have subpopulations of the T helper cell. T helper cell 1 will actually produce tumor necrosis factor and interferon gamma that will stimulate more macrophages, and this can cause delayed hypersensitivity reactions. T helper cell 2 will produce interleukin 4 and other B cell growth factors that will actually activate B lymphocytes to become plasma cells that produce antibodies to the antigen that was presented. Some of these CD4 cells or helper cells will become memory cells. So they just kind of sit over on the sidelines and go, you know what, you guys are doing okay. I don't need to do anything right now. The T helper 17 cell is going to actually secrete a number of pro-inflammatory mediators, increasing inflammation. And some of these CD4 cells will actually become regulatory T cell that will dampen the immune response as appropriate. Also, through a number of different cytokines, we can actually stimulate CD8 cells. But primarily, the CD8 or cytotoxic T lymphocyte will be activated when an antigen is presented from an APC on the MHC1 major histocompatibility antigen. This then will activate the CD8 receptor that will then activate the CD cells to become activated to the antigen. Some will actually undergo clonal expansion to become memory cells, and some will actually be activated into active cytotoxic T cells. When the T cell receptor recognizes the antigen on, let's say, this infected host cell, it will bind to that cell and release a couple of products. One of these will be perforins. Perforins will actually punch holes in the cell membrane of the infected host cell attached to the cytotoxic T cell. It also will produce granzymes that will enter through those perforations and actually destroy this infected host cell. So this is just a table showing the functions and major important features of each of these cells. I'm not going to talk about the gamma delta T cells because they do not respond to specific antigens, but they do respond to specific antigens, but also respond to non-specific markers. Once the B lymphocyte is stimulated, again, it will produce then antibodies, but another function of the B cell is if the B cell's receptor is present and this microbe has the correct antigen, it actually will bind to the B cell receptor itself, and this can activate the B cell to become plasma cells. But it could be that actually the B cell then will act as an antigen presenting cell and present that antigen to a helper T cell, just like a macrophage or dendritic cell. Once the B cell is activated, we then see the differentiation into the B memory cells that will remember this, into the active antibody secreting plasma cells, and into some regulatory B cells. Regulatory B cells will secrete interleukin 10 that also helps regulate the T cell response. So stimulated T cells produce a large population of genetically identical daughter cells, and what other type of cells? True or false? B cells can only be activated by T helper cells. CD8 cytotoxic T cells are activated by antigen complexed with what molecule? And plasma cells can produce about how many antibody molecules per second? And again, you may have to do some research on that last one. So the T cell response, cell-mediated immunity. This requires direct involvement of the T lymphocyte throughout the course of the reaction with the antigen-bearing cell. The T cell response is amongst the most complex and diverse in the immune system, and I'll be real honest, we still don't fully understand it even after years of studying it. It involves several subtypes of T cells whose action are dictated by the antigen-presenting cells that activate them. T cells require some type of major histocompatibility recognition before they become activated to their active form. All T cells produce a number of different cytokines that have a broad spectrum of biological effects. So let's look a little closer at the T helper cell. I said these were the generals of the immune response, and they literally play a central role in regulating the immune response to antigens. This will include activating B lymphocytes and other T cells. They are involved in activating other macrophages, either directly by receptor contact or indirectly by releasing cytokines such as gamma interferon. Stimulation of these T helper cells that contain the CD4 receptor by the antigen major histocompatibility complex 2 differentiates them into, again, the T helper 1s, T helper 2s, or T helper 17s. Depends on what type of cytokines the APCs actually secrete will determine which one of these that the activated T cell will differentiate into. Regulatory T cells in that T helper cell class will also carry CD4 markers, and they will actually help control the inflammatory response. The regulatory T cells are specifically involved as well in preventing autoimmunity. That's why we think perhaps that part of the reason autoimmune diseases develop is that we have lost the regulatory T cell function in some way. These regulatory T cells make sure that the immune response does not inappropriately target normal microbiota or self cells. The regulatory B cells also regulate the degree of response from T cells. So once B cells become activated to become plasma cells by cytokines from activated T cells, the regulatory cells will help act to regulate them. And already activated regulatory B cells can secrete their own cytokines to actually dampen the T cell response. Now, cytotoxic T cells, these I call the special ops units because these are the ones that will go out directly and interact with an antigen-bearing substance and kill it. So cytotoxicity is the capacity of these cytotoxic T cells to kill a specifically targeted cell. The killer cytotoxic T cell carries the CD8 receptor and recognizes the class 1 MHC proteins. So it must recognize a, quote, foreign, and really I want to put in here non-self antigen complexed with one of these MHC1s. The cytotoxic T cells again release perforins that are proteins that will punch a hole in the membrane of the antigen-bearing target cell. And this will cause ions to leak out of the target cell, eventually killing them on their own. And it can create a passage for the other thing that cytotoxic T cells produce, and those are granzymes. The granzymes are enzymes that will dissolve and attack proteins within the target cell. And after attack with perforins and granzymes, the targeted cells then go through the death process by apoptosis. Cells that are targeted by the cytotoxic T cells include virally infected cells and cancer cells, and cells from other animals or humans. And these are actually cytotoxic T cells binding to this targeted cell, and it will be releasing the perforin and granzymes, and you'll notice already that there are some blebs that are being created here in our targeted cell, and that's part of our apoptosis. Now, I said I wouldn't really mention the gamma delta T cells, but they are distinct from other T cells in the fact that they have T cells rearranged to recognize a much wider range of antigen, and they can even respond to certain PAMPs on microorganisms. So they can actually respond fairly quickly and do produce memory cells when they are activated. The natural killer, or NK, or sometimes referred to as null cells. These are lymphocytes that are related to T lymphocytes. We used to call the natural killer cells a non-T, non-B cell, but actually we now think that probably they are a highly modified T lymphocyte. They lack specificity for antigens, so they don't have the T cell receptors. They do circulate throughout the spleen, blood, and lungs, and they are literally our immune surveillance system. First killer cells to attack cancer cells and virally infected cells in a similar manner to the cytotoxic T cells. But the NK cells have not been activated specifically through a CD8 receptor and MHC1 class antibody or 1 class marker. So they're not considered to be a part of the specific cell-mediated immunity because they do not possess those T cell receptors for the antigens. The natural killer T cells, which are a little different, were recently discovered, and they share properties of both T lymphocytes and the natural killer cells themselves. They do display T cell receptors and receptors or markers found on the natural killer cells. They typically will be stimulated by glycolipids on foreign cells, and they rapidly then will produce cytokines, granzymes, and perforins. They will trigger apoptosis in the target cells. So can you describe the functions of the T helpers 1, 2, and 17 cells? Cells that regulate T cell activities are known as what? Cytotoxic T cells use what two chemicals to destroy virally infected cells or cancer cells? And blank have similar functions to cytotoxic T cells. Now, let's turn our attention to the humoral response, those that are actually seen under control of the B lymphocyte. And again, activated B lymphocytes will be activated to the antibody producing plasma cell. The antibody or the immunoglobulin protein. So, four polypeptide chains connected by disulfide bonds are what will create the immunoglobulin or antibody protein. There are two functionally distinct fragments. The antigen binding fragments, or the FAB regions, and these are on the upper arms of the immunoglobulin molecule, and the crystallizable, or what's now called the constant fragment, the FC fragment, which is the rest of the molecule. So we see here our four polypeptide chains. We have two short chains and two heavy chains. The two light chains are bound to the heavy chains by disulfide bonds, and the two heavy chains are bound together by a double disulfide bond. They may have carbohydrates added to, especially the FC region, or they may have some other things as well. But they basically form this kind of Y-shaped structure. The FAB region is basically the upper part of the arm, and this actually will be or will consist of what's known as the variable region. The FAB region is what will bind to the antigen, hence the antigen binding fraction. The FC region, the amino acid sequence within the FC region does not vary, and so this will actually be what will be identifying our class of antibodies. So the FAB fragment is the amino terminal end that consists of the variable regions of the heavy and light chains. It's a specialized region of attachment between the FAB region and the FC region that allows swiveling of the FAB fragment, so the arms can actually move. The FC fragment identifies a specific class of antibody, but it can also serve as an anchor if this antibody is actually embedded into the surface of a cell. So it's involved in binding to various cells and molecules of the immune system itself. The hypervariable region is the site on the antibody where the antigenic epitope will bind. Here, the amino acid content is extremely variable, and it's based specifically upon which antigen this antibody was made to. A minimal complementary fit is necessary for the antigen to be held effectively to the antibody. So in this picture, we see a perfect fit between the antigen and the hypovariable region of the FAB region. So it's a perfect fit, and we get a very strong activation of the antigen-antibody complex. Whereas this one will not fit at all into the amino acid sequence, and so there's no activation of that antibody. And this one we have a kind of it almost fits but not quite, and so we get a very weak activation of the antigen-antibody complex. The antigen-antibody interaction is the principal activity of the antibody, and this is to unite with, immobilize, call attention to, or otherwise neutralize the antigen for which the antibody was formed. And there are several different ways that the antigen-antibody complex can work. One is opsonization. An organism that has numerous antigens on its surface, when it's bound to its antibody, it's literally like the antibodies coat this foreign substance, and this coating will actually highlight the cell for phagocytosis. Agglutination, we could actually clump up the cells that are presenting the antigen. Neutralization and precipitation. Neutralization, we bind to the antigen, and this may stop the antigen from binding to another type of receptor. And this is very important also in viruses, in the fact that if the antibody binds to the virus, oftentimes the antigen of the virus is what needed to attach to a cell receptor for adsorption. And since viruses can be so small, they can actually become soluble in body fluids. But if I attach this big old honking antibody to it, it will actually cause it to precipitate out of solution and not be able to bind to its host cell. Some antibodies actually will act as antitoxins when bound to the antigen. And the antigen-antibody complex is the classical pathway of activation for the complement system within the blood. So here's some pictures. So here's a bacterium, and these antibodies have bound to the antigen on the surface of the bacterium, coating the surface of the bacterium. And especially macrophages see these coatings of antibodies as big neon signs that say, "Eat me, I'm yummy." And so this is the process of opsonization. Again, neutralization, antibodies block the binding of these, let's say, viral spikes to their receptor on their host cell, thereby blocking the adsorption of the virus into the host cell. Or again, it may be that this virus is very, very small and soluble, but when we add all these antibodies, it actually causes precipitation of the virus out of solution. Here we see agglutination. So this antibody can bind to this cell and this cell through antigens and actually cause clumping. You've probably done this with blood typing plates. Antitoxins, if the microorganism is producing a toxin, antibodies can actually be made to that toxin, being that it's a foreign substance, and then the antibody binds to that, blocking the function of the toxin. And antigen antibody complexes can activate complement that then form those membrane attack complexes that will then punch holes in these foreign cells and cause them to lyse. The Fc region of the antibody can actually enhance opsonization. So the attachment of the antibody to the foreign cell exposes these Fc region fragments to phagocytes. And this is really like the Fc region is carrying that neon sign that says, "Eat me." Certain antibodies have regions on the Fc region that actually bind complement. In other cells, binding of the Fc region causes the release of cytokines from the cell that it's bound to. For example, the Fc region of IgE class antibodies will actually cause the release of histamine from basophils and mast cells when the antigen binds to it. And in fact, most of the IgE class of antibody is actually found embedded into the cell membrane of mast cells or basophils. Now, varying amounts of carbohydrates are oftentimes attached to the constant regions of the different classes. A J chain joins the monomers of IgA and IgM class antibodies. The secretory component actually helps IgA move across mucous membranes into the mucous fluids. So let's look at our 5 classes. I little g stands for immunoglobulin, and the capitalized letter stands for the class. So IgG, this is the largest number of circulating antibodies that we have in the body right now. It consists of a single monomer protein with 2 antigen binding fragments, and we call that the valence of the antibody. So they make up about 80% of circulating antibodies. They are the longest lived, having a half-life of about 23 days. A couple of unique things about IgG is one, this is a class of antibody that will cross from the maternal blood into the fetal blood. And why might this be a good thing? And why might it be a bad thing? IgG is very good, in fact, the only class that actually fixes complement, starting the complement cascade. And the Fc region binds very well to phagocytes. So one of the major functions of IgG is opsonization. Biological functions, it is long-term immunity. In fact, most of the circulating antibodies, again, in your bloodstream have been made to things you've been exposed to, and they typically stay in the blood for a fairly long period of time where they can be activated. They actually are sometimes known as the memory antibodies because they are so long-lived. They neutralize toxins. Again, opsonization is one of their big things, and fixing or starting complement. Now, IgA. IgA can exist as a monomer, and primarily this would be in bloodstream, but they only make up about 13% of circulating antibodies in the serum. Most of the time, IgA, when it is made, will be secreted across mucous membranes into the mucoid solutions, where they then are bound together with another IgA, and this is known then as a dimer. The dimer is created by that J chain. So where we find most IgA is in body secretions, saliva, mucus, tears, breast milk. And so IgA actually is going to be important in the newborn in that it will help establish some of the immunity of the newborn, especially in the digestive system. IgM. IgM exists as a pentamer. There are 5 of the antibody molecules joined together by a J chain. This is the largest in size of all of the classes. However, it only makes up about 6% of circulating antibody. And one of the reasons for this is it's typically used up very quickly. The average life or half-life in the serum is about 5 days. So it is actually very large in size and can bind lots and lots of antigen with a valence of 10 here. And I will say this, it is always the first class that is produced to an antigen. It will be used up quickly, but it will always be the first class produced by plasma cells that have become activated. They may serve as B cell receptors, but that's actually going to be another class that's primarily going to be that. And I should have said, IgG is not the only one that fixes complements. IgM does as well. And that's a good thing since it is the first one being produced. Now, IgD and IgE actually are found in very small numbers within the bloodstream. That's because they both have unique functions. They both exist as monomers, and what we'll see is that they actually have unique functions upon a couple of different cells. IgD acts as the B cell receptors. The Fc region will embed into the cell membrane of the B lymphocyte, and the antigen binding fractions then will be out there so that if an antigen binds, that B cell will now be activated to that antigen again. IgE is actually going to be found on the surface of mast cells and basophils. IgE on mast cells, when the antigen binds to those embedded IgE, will cause the mast cell to actually degranulate. And if we get too many of those IgE-coated mast cells degranulating, we can actually then have hypersensitivity reactions. Also, IgE is very important with eosinophils in the destruction of helminth infestations. Now, how can I monitor antibody production over time? Well, we do this with something known as the titer, and this is simply the concentration of antibodies to a specific antigen in the serum. This can be measured over time to determine how the immune system is reacting to the antigen. Now, I can measure other things, but since the antibodies are in the bloodstream and accessible, this is a very easy thing I can actually measure to determine how the immune system is responding. So I can see here that days after onset, so I had exposure here to my antigen. And I'll see then that I get an antibody response. And again, IgM is always going to be first, usually followed by IgG, and then the rest of them will be produced by other plasma cells. Now, the primary immune response. This is the immune response that occurs the first time we see an antigen. And this actually takes some time in order to gear up. We actually see a latent period from time of exposure to the time that we see antibody production of somewhere between 5 to 10 days. The reason for this is that we have to do the front end processing. The antigen presenting cell has to engulf the antigen bearing substance, digest it, process the antigen, put it on an MHC protein, present it to the T helper cell. The T helper cell then has to become activated, undergoing clonal expansion and releasing cytokines that will activate the B lymphocyte. The B lymphocyte then has to undergo its clonal expansion to the plasma cell. The plasma cell has to gear up for protein production. And that all of that front end processing then takes somewhere between about 5 to 10 days. On average, usually about 5 to 7 days. And so this is a period that's marked by a lack of antibody production because all of this front end processing of antigen presentation, T helper cell activation, B cell activation to plasma cells takes a period of time. The antigen is concentrated at this time really in lymphoid tissue, and all of this front end processing is occurring. As plasma cells then gear up, they begin to produce the antibodies, and the serum titer increases, usually to a plateau. The reason for that is because we are going to start using these to attack whatever the antigen-bearing substance is. And so antibody levels will then taper off to a low level over several weeks to months. Secondary immune responses, or the amnestatic response, occurs when the immune system is exposed again or subsequently to the same antigen weeks, months, or sometimes even years later. The rate of antibody synthesis, peak titer, and length of antibody response is greatly increased over that of the primary response. The reason for that is in that primary response, what did we make? We made memory cells. So our primary response, again, our latent period from time of exposure to the time that we start to see the rise of antibodies is somewhere between about 5 to 10 days. The first antibody produced will be IgM, and it will be used up very quickly, followed by IgG. So the overall total will come up here and decrease to usually not zero, but to at least low levels. Then, when we are exposed subsequently to that antigen a second time, we only have a latent period of basically 1 to 2 days. That's because we're going to see a rapid rise in both IgM and IgG because of the production of memory cells in the primary response. You'll also note that the IgG actually stays up higher this time because we think, oh gosh, if we've seen it twice now, we might see it a third time. So why not just keep the levels of IgG high so that even though, again, we're still going to see front-end processing and activation of more B cells to this antigen, but the IgG will just be in the blood to actually start dealing with it early. So why do we give vaccines? And why do we oftentimes give a series of vaccines to begin with? Well, here's the reason. We hope that the first time we give that vaccine, we get a primary immune response. Now, again, our antibody levels typically fall pretty quickly. But what did we do? We created memory cells on both the T and B cell side, so that when we get that second booster, or we see the disease for real, will we have a faster immune response? Yeah, our lag time's only now going to be 1 to 2 days, and so we're going to get a much higher response more quickly. So what's the likelihood if you saw this a third time, and let's say this time you were actually exposed to the real disease, what would you see? Well, you would actually see a much quicker response, and therefore, sorry, the dog barked, and therefore we're probably not going to get sick. So, coating of pathogens with antibodies is known as what? Antibodies secreted in the tears, saliva, and mucous membranes are of what class of antibody? And the first class of antibody always to be secreted, especially in a primary immune response, is going to be what class? The most prevalent antibody class and the only class to cross the placental barrier is what? And the blank response of the immune system provides a large concentration of antibodies in a very short period of time. So we can actually divide immunity into active and passive immunity. Active immunity means your immune system responds to the antigen. So we mount a response. And types will be naturally acquired active immunity and artificially acquired active immunity. An example of naturally acquired active immunity is you get the disease and you get sick and you recover. Artificially acquired active immunity would be an example, or one example would be a vaccine. Now, passive immunity is a little different. Passive immunity, it means that we're going to give protective components of the immune system, but your immune system did not make them. Someone else's immunity did. So the person receives protective entities, either antibodies or sensitized T cells made by another's immune system. And again, we have both a naturally acquired passive immunity and artificially acquired passive immunity. A good example of naturally acquired passive immunity is maternal antibodies, either IgG through the placenta or IgA through milk. Artificially acquired passive immunity is the one you're probably least familiar with, and this is when we would give something like an immunoglobulin injection. The immunoglobulins antibodies were made by someone else's immune system or B cells in culture, but we're giving them to you for protection. So your immune system didn't make them. Passive immunity will always be temporary, while active immunity is typically fairly long or sometimes can even be lifelong for the individual. So natural immunity acquired through normal life experience. The active form, again, is simply getting the disease and recovering from it. Passive naturally acquired immunity is going to be maternal antibodies. Artificial immunity, the active form with vaccination. Does your immune system make an immune response? Well, we hope so. That's why we're giving you the vaccination. And then passive immunity, passive immunotherapy involves preparations that contain either specific antibodies or sensitized T cells that we give to the patient. So we can actually use this artificial passive immunization as an immunotherapy. And this is actually known as intravenous immune globulin therapy, or IVIG. It's also sometimes known as gamma globulin therapy because the other name for antibodies is gamma globulins. So we extract these antibodies from pooled blood of human donors. And then the IVIGs are concentrated to increase the potency and eliminate any potential pathogens. Most of these IVIGs are injected intramuscularly, and the protection usually is for about 2 to 3 months. A really good example of this is actually immunoglobulins to hepatitis A antigens. So when we have hepatitis A outbreaks, we will go in and, quote, vaccinate, even though it's not a true vaccination, but we will give IVIGs to Hep A antigens to individuals that could be potentially exposed. And again, usually those antibody levels will last somewhere between about 3 weeks to about 3 months. So IVIGs can actually be derived from a more defined group of donors who are convalescing in a hyperimmune state after certain infections. So one way that we treated some of the Ebola patients was actually the doctor that was treating Ebola patients in Africa, came down with Ebola, and was brought back to the United States. We actually took his blood and extracted the antibodies that were made to the Ebola antigen. And we used that concentrated serum with his antibodies to treat other patients that had Ebola. So these IVIGs will contain high titers of specific antibodies obtained from a smaller pool of patients. And in fact, in that case, it was a single patient. So it's useful in prophylaxis in patients who have been exposed or could potentially have been exposed. But because oftentimes the pool is very small, the amount of these IVIGs can actually be limited. Now, vaccination causing an artificial active immunity is exposing the person to a material that is antigenic but not pathogenic. And the discovery of vaccination was one of the farthest reaching and probably most important developments in medical science. And again, Edward Jenner in late 1700s was the first one that actually created a, quote, true vaccine in the fact that he used cowpox to actually vaccinate against smallpox. And so there was some cross reactivity, and so the person that received the cowpox lesions actually developed antibodies that also bound to smallpox antigens. The principles of vaccinations is that the vaccination will stimulate a primary immune response that primes the immune system, making memory cells for future exposure to a potential virulent pathogen. If the pathogen then later enters the body, the secondary immune response will be almost immediate, again within 24 to 48 hours, very powerful and oftentimes sustained. So, when we make vaccines, what do we have to consider? Well, one, are we choosing the correct antigen? How effective is the antigen going to be at producing an immune response in the patient that receives the vaccination? How about ease in administration? What about safety concerns? And this is probably one of the biggest issues that we have in vaccination considerations. And certainly the cost. So here's a checklist for an effective vaccine. It should have low levels of adverse side effects or toxicity and not cause serious harm. Now, we used to actually, quote, vaccinate for smallpox in a process known as variolization. Variolization was actually practiced 2,000 years plus before Edward Jenner came along. And what the Chinese would do is they would take lesions from a smallpox patient and actually give it to a non-infected patient. So the patient either developed immunity, got the disease, survived, and had lifelong immunity, did not get the disease because they just simply were already immune to the disease, or they got the disease and died, and so we didn't count them in the population anymore. So, considering the fact, though, that smallpox kills 2/3 of the patients that got it, this was not really a very good method of protecting other people. Vaccines should protect against exposure to natural wild forms of the pathogen. It should stimulate both an humoral and cell-mediated response. It should give us at least fairly long-term lasting effects, producing memory, and it should not require numerous doses or boosters. The vaccine should be inexpensive, have a relatively long shelf life, and be easy to administer. So, what are some of the vaccine preparations? Well, we can use whole cell or whole viruses that have been modified and they are still living or active, or what we call attenuated. We've tweaked them genetically to decrease pathogenicity. Typically, they confer longer lasting protection because they are going to enhance the response because they are still living or active. Usually requires fewer doses or boosters than other types of vaccines. They are particularly effective at inducing cell-mediated immunity. But the problem is sometimes these could remutate back to the pathogenic form. Killed cells or inactivated virus vaccines, this we've actually killed the organisms or inactivated the virus. Part of the organism preparations are becoming more common. And with part of the organism preparations, what we do is we extract the antigenic molecules derived from the bacterial cell or viruses and use just the antigenic portion, not the entire either attenuated or killed cell or virus. Subunit vaccines, so these are what these part of the organisms are, is again, we simply will take the antigenic portion. And we can do this in several ways. We could actually just clip off the antigenic portion themselves. Okay, or we could take out the part of the organism of the bacterial cell, say for example. Or we could actually take the genetic that produces the antigen from the pathogenic organism and genetically engineer non-pathogenic organisms to create that protein or toxin for us. And then we can actually use that as the antigen in the vaccine. Toxoids are actually inactivated or attenuated toxins. So we don't get the toxic part, but we get the part of the toxin that is antigenically active. These viruses are actually usually much safer because we're not giving any extra stuff. that would come along with whatever the antigens are in the whole cell or virus. This just shows whole cell or talks about whole cell vaccines. And again, the killed vaccines, we heat or kill the antigen or inactivate the or virus and administer those directly. The pathogen cannot multiply. Does it stimulate immunity? That may actually be something that we may not know fully. Okay. Live attenuated cells or viruses, this is where the virulence then is decreased, so they are still living or active, but they have the same antigenicity as the true pathogenic organism. We can actually see multiple disease vaccines that have been developed, and probably some of them that you've heard about is DTaP, which contains diphtheria, pertussis, and tetanus. And this is the primary one that we give now for tetanus because we figure you probably need a booster for diphtheria and whooping cough at the same time. We have some that contain several different ones like Pediarex that contains diphtheria, pertussis, tetanus, Hep B, and poliovirus all in the same. Twinrix contains hepatitis A and hepatitis B. The measles, mumps, and rubella vaccine. This is the MMR that we've seen for many, many years. And then ProQuad actually has the MMR and includes herpes varicella zoster for chickenpox. Dozens of bacterial, viral, protozoan, and fungal diseases still remain without a functional vaccine, such as malaria, HIV that causes AIDS, various diarrheal diseases, respiratory diseases, worm infestations. And I'll say, we probably have more bacterial vaccines than we do virus, protozoan, and fungal vaccines. Most existing vaccines are out of the reach for much of the world's population. And so while we have, quote, eradicated smallpox through vaccinations in the world, we are now targeting polio as to be the second disease, quote, eradicated from the world. And primarily, we're looking at DNA recombinant vaccines. So these are, one, much safer. The microbial DNA of the virulent organism is inserted into a plasmid vector and inoculated into a recipient cell. The recipient cell is either then used as the vaccine because it's non-pathogenic, or we'll let the recipient cell actually then make the plasma or make the protein that's the antigen. Human cells then can take up the plasmid and express the microbial DNA in the form of proteins, and this then will actually allow the human cell to actually produce the antigenic protein of the organism, and the immune system can respond to it. So just simply shows we took the gene from the organism, put it into a plasmid, injected it into the human, allowed the human cells to take up this plasmid, and hopefully then the antigenic protein would be made from that or from the gene, and the human cell then would express the antigen of this bacterium, and the immune system then would respond to this antigenic molecule on the surface of this cell to destroy this now, quote, foreign cell. Traditional routes of vaccine administration include subcutaneous injections, intramuscular, and even intradermal. Oral or nasal vaccines are simply applied to the mucosal surface of the nasal or oral cavity, and they are available for only a very few diseases out there. Now, oftentimes we are going to add things to the vaccines known as adjuvants. These are special binding substances that are added to some vaccine that will couple to the antigenic substance and actually enhance the immunogenicity. That's why when I asked the question to an epidemiologist and another microbiologist why our inactivated flu vaccines are effective at all, they're saying that, well, they're hoping that the adjuvant bound to the flu antigen will cause some sort of immune response. The adjuvant precipitates the antigen and holds it in the tissues so that it will be gradually released, and so we get a little longer exposure time as well. This will facilitate contact with the antigen presenting cells and hopefully cause more of a response to the vaccine. The most common adjuvant that we see in vaccines is alum, which is an aluminum hydroxide salt, but there are also some of the heavy metals such as mercury and silver that are used. Now, common side effects of vaccine, local reactions at the injection site, because we are putting a foreign substance in. We can see systemic fever, and we can also see allergic reactions. And this is actually a dog that was given a vaccine, and a few hours later, they broke out in hives. And so this can actually be seen in humans as well. Relatively rare side effects include panencephalitis. 1 in 220,000 or so vaccinations from the measles vaccine will actually result in this severe side effect of encephalitis in these individuals. Back mutations to a virulent strain, especially with those attenuated or modified live vaccines, such as the polio vaccine. We may actually have seen a back mutation to a more virulent or to a virulent strain of polio in the last few years, as we've seen actual polio cases or polio-like cases in the United States. And a disease due to contamination with dangerous viruses or chemicals that were simply in the vaccine as a contaminant product. Neurologic effects of unknown causes has been associated with pertussis and swine flu vaccines. And allergic reactions to egg or tissue cultures that some of these vaccines have actually been grown from. So we're very concerned about vaccine safety, and some people have attempted to link childhood vaccinations with the development of certain diseases, including diabetes, autism, and even asthma. And there's been a very public debate over vaccine safety. It is difficult for parents and consumers to discriminate oftentimes between good and bad information that is out there in the world. Scientists are trying to address parental fears and provide reliable information. And so one of the biggest cases was the link between the MMR, the measles, mumps, rubella vaccine, and autism. The Institute of Medicine in 2011 published the results of a comprehensive examination of childhood vaccines and stated unequivocally that the MMR vaccine does not cause autism. And so this has been completely disproven. The MMR vaccine does include a tiny bit of mercury as an adjuvant, and this is what many people thought might have been causing the autism. But there has been absolutely no link between autism and the MMR vaccine. Results of unfounded vaccine fears. We've seen outbreaks of measles, mumps, diphtheria, polio, even typhoid fever and whooping cough all over the United States due to unvaccinated children. And these can be especially deadly even in neonates. We've seen some of these outbreaks in college dormitories, and this could actually be because some of these vaccines actually need boosters in later life, especially in the late teens. Anti-vaccination religious communities. We've seen outbreaks of these diseases. Now, one of the things that we would like to see from vaccination protocols is something known as herd immunity. Each microorganism requires a certain density of susceptible individuals in the population or the herd to maintain the chain of transmission and immunity against a particular organism. With sufficient numbers of immune individuals, the microbe will not spread. Global immunity through mass immunization confers indirect protection on non-immune members. So because the organism is not being transmitted within the population, those that are not immunized or do not have protection will be protected through those that do. So maintenance of herd immunity through immunization prevents epidemics. So vaccinations are traditionally most prominent in childhood because we want to stimulate the immune system when it is absolutely the most active, and that's during childhood. However, adults do also need vaccinations. We need to boost older immunizations that we saw in childhood or to protect against adult infections such as pneumonia or shingles. And it can provide special protections in people with certain medical conditions. So these next few slides are just simply recommendations from the CDC of which vaccines and at what age vaccines should be given. This I don't think hasn't changed much since 2016. So within the first 2 months of life, hepatitis B with a booster, usually between 4 to 16 months after that. We need our first DTaP. And hepatitis, or sorry, Haemophilus influenza, PVC, IPV, and influenza, the MMR, chickenpox vaccine, and hepatitis A. This is the recommendation immunization for children between 7 to 8 years of age, and you can just look through those. And then the recommended adult immunization. And by no means am I advocating that everybody should get every one of these vaccines, but it is something that you need to be informed about and actually look at with a discerning eye. So breastfeeding would be an example of what type of immunity. Vaccination is an example of blank blank immunity. Vaccines are either blank organism or part of organism preparation. True or false, there are vaccines against all major infectious diseases. And blank or blank vaccines stimulate IgA productions on mucous membranes. And true or false, there is no link between the MMR vaccine and autism. So that does it for our chapters within the text. And what we're going to do with the next 3 lectures is actually look at some selected diseases that will be found in the rest of the chapters of the book. And so we'll look at those when we get into the Disease 1 lecture. Thanks. Have a great day.